So, hello everyone. From today onwards, I'm starting microbial fermentation and I'm going to cover the topics in 2 to 3 lectures. So, let us begin with Introduction to Microbial Fermentation. So, looking at the simple definition of microbial fermentation, is that it is a metabolic process of breakdown of complex molecules into simpler ones by microorganisms, and in this process, energy is extracted, which is used by the microorganism in order to maintain its form. Then other simple definition is that fermentation is an extraction of energy from carbohydrates or macromolecules in the absence of oxygen. So in the absence of oxygen also, the process is called fermentation. Like for example, RBCs and active muscle cells. So but we are interested in microbial fermentation. So for us, be it for microbial fermentation, be it an anaerobic organism or aerobic organism, it is the metabolic process where complex molecules are broken down into simpler ones and energy is extracted. So metabolic process in anaerobic or aerobic microorganism is called microbial fermentation. Moving further, coming over to industrial microbiology, so it is a field where commercial products are produced by microorganisms on large scale. So basically, in industrial microbiology or industrial fermentation, microorganisms are exploited for the production of certain products of interest. So, what are the common products of industrial microbiology? Well, there are many products, but some of them are antibiotics like penicillin, tetracycline, and others. These are produced by industrial fermentation. In antibiotics, the natural antibiotics and semi-synthetic antibiotics are produced by industrial fermentation, but not the synthetic ones. Synthetic ones are produced in labs. Then there are certain enzymes like glucose isomerase, laundry proteases, and lipases. So these are also produced by industrial fermentation. Then there are food additives like vitamins, amino acids. These are produced. Then chemicals such as the citric acid, vinegar, biofuels such as alcohol and biodiesel. These are the products of industrial fermentation. Then alcoholic beverages like beer, wine, distilled spirits, vodka and all. These are produced by industrial microbiology. So these are some common products, but there are other products also like insulin. These days, insulin which we use, this is also a product of industrial microbiology, and there are so many. So moving further, now organisms which are most commonly used in industrial microbiology are fungi, that is yeast and mold, and prokaryotes such as streptomyces. So these are the microorganisms. Now industrial microorganisms or industrial strains are metabolic specialists capable of synthesizing one or more products in high yield. So industrial strains are the special strains, and these are different from the wild type strains. So what do microbiologists do, or any scientist working in the field do? They look for microorganism, the useful microorganism which has the ability to produce the useful product from nature. Okay, and once they get to find out the microorganism, the useful microorganism which has the ability to produce the useful product, now they have a task to convert this wild type microorganism into the industrial strain, which is a high yielding strain, because high yield is very important for commercial production, and for any product making company, yield is very important. Yield is at the center point because yield means profit. Okay, so the scientist, they have to manipulate the wild type strain in such a way that it can produce a product of interest in higher yield. Next, Now let us look at production and scale. What is production and scale? So the microbial growth is described into three phases. Everybody know about the lag phase, exponential phase, and stationary phase. Okay, this is a growth curve. Okay, on y-axis there is number of bacterial cells, on x-axis there is time, and you can see that every microorganism has a growth curve which is composed of three phases: lag phase, this is a smaller lag phase, and there is a log phase which is also called exponential phase, then there is a stationary phase, and then after stationary phase cells they die. Now the microbial products, they are of two types: primary and secondary. The primary products, they are produced in the log phase. Okay, the exponential phase. Primary products are produced in the log phase, and the secondary products are produced in the end of the log phase and in the stationary phase. Okay, the example of primary product is alcohol, and the example of secondary product is antibiotic. Okay, these are the most common examples. Now, in order to develop a commercial microbial product, there are two things are important. One is that the bioprocess is developed in small scale, and in that industrial strain is also developed. Then after that, once the bioprocess is developed in small scale, then this bioprocess is transferred in large scale for commercial production, and this transfer of bioprocess in large scale is also called scale up. Now what is bioprocess? It is nothing but it is a process of producing microbial product in higher yield for commercial production. So basically it is a process which is used or applied for producing the commercial product in higher yield. So what process that the scientist or industrial microbiologist have to apply so that the product is produced in higher state. So this is called in higher yield. So this is called bioprocess. See, for the commercial production of any microbial product, there are few steps which are to be taken, and so I'm going to discuss those few steps in a broader manner which are followed for the final commercial production of the product, microbial product. So I have taken the example of a novel antibiotic. Suppose a novel antibiotic has to be commercially produced. What route it would follow for its production? So first thing would be isolation and screening of antibiotic producing strain from the nature. So first step is that the novel antibiotic producing strain would be screened and isolated from the nature. Okay, and screened for its antibiotic producing capability. First thing. Then the novel antibiotic will be extracted and purified and would be tested for their effectiveness and toxicity in animals, on animals. Okay, so second step would be testing of the effectiveness and toxicity in the lab on animals. Okay, so once found very effective and very less toxic, the antibiotic would be then tested on human beings. Okay, so third step would be clinical trials, that is testing of the novel antibiotic on human beings. Okay, after the successful clinical trials, so clinical trials happen in phase 1, phase 2, phase 3, and then after that, if the antibiotic passes through these clinical trials, then it goes for approval from any government agency. In USA it is FDA, in India it is DCGI. Okay, then the novel antibiotic goes for approval from FDA or any other agency. Once the FDA approves the antibiotic, then it goes for commercial production. So these are the few very basic steps which are followed for the commercial production of any novel antibiotic. Okay. सी जो ड्रग मेकिंग कंपनीज होती हैं उनकी अपनी R&D यूनिट होती है जिसमें कि ये सब काम जो है वो उनके अपने साइंटिस्ट होते हैं जो ये सब काम करते हैं. ठीक है. तो उसमें क्या होता है कि जैसे एक बार अगर उनको एक नोवल एंटीबायोटिक मिल गया, ठीक है, उसकी वो सारी स्ट्रक्चरल स्टडीज़ कर लेते हैं और फिर एनिमल्स पे जब स्टडी करते हैं और जब नोवल एंटीबायोटिक जो है इट प्रूव्स टू बी वेरी इफ़ेक्टिव एंड वेरी लेस टॉक्सिक, तो उसके बाद फिर उनको लगता है कि ठीक है प्रॉमिसिंग है ये कंपाउंड, तो आगे फ्यूचर में इसका प्रोडक्शन हो सकता है. तो जैसे वो क्लिनिकल ट्रायल्स करते हैं, क्लिनिकल ट्रायल्स में बहुत टाइम लगता है, ठीक है, इसमें कई साल लग जाते हैं. तो मीनवाइल जब क्लिनिकल ट्रायल चल रहे होते हैं साथ ही साथ वो उसकी कमर्शियल प्रोडक्शन पे भी काम शुरू कर देते हैं. ठीक है. यानी कि जो बायो प्रोसेस डेवलप करनी होती है उन्होंने वो साथ ही साथ शुरू कर देते हैं. ठीक है. जैसे ही क्लिनिकल ट्रायल्स सक्सेसफुल हो गए उसके बाद फिर उसका कमर्शियल प्रोडक्शन जो है वो शुरू कर सकते हैं. ओके. Product yield is a central issue with virtually all pharmaceuticals, and even after commercial production of an antibiotic has begun, research often continues to obtain higher yielding strains or a more efficient process. So, product yield is at the center for all the pharmaceutical companies, and so every pharmaceutical company has a research and development unit, and the researchers they keep on working on developing the strain for higher and higher yield. So they keep on working on the development of the strain as well as the bioprocess for higher yield. Now in this lecture I will discuss industrial production of penicillin and tetracycline. So starting with penicillin first. So penicillins come under the class of beta-lactam antibiotics because of the beta-lactam ring present. So all penicillin molecules they have beta-lactam ring in them. So what is this beta-lactam ring? Now look at this diagram, this figure or this structure. This is 6-amino penicillinic acid. It is a parent compound which is there in all penicillins. Okay, so this compound has a beta-lactam ring encircled. यहाँ पे जो circle है इसके अंदर है, this is a beta-lactam ring. ठीक है, this is a 4-membered ring and this ring is present in all the penicillin molecules, and that is why it is called, all the penicillin molecules are called beta-lactam molecules, like they come in the category or in the class of beta-lactam antibiotics. Apart from penicillins, cephalosporins are also beta-lactam antibiotics. Let me check once. So penicillins are produced by fungi of genera Penicillium and Aspergillus. For commercial production of penicillins, high yielding strains of the mold Penicillium Chrysogenum is used. Okay, so remember Penicillium Chrysogenum is a fungi which is used for commercial production of penicillin. So 3 kinds of penicillins are produced, and these are biosynthetic penicillins, natural penicillins and semi-synthetic penicillins. So first let us first have a look on the structure of penicillin molecule. So this 6-amino penicillinic acid, this is a parent structure. Okay, this is a parent compound and this is composed of a thiazolidine ring. Okay, this is a thiazolidine ring, this 5-membered ring. Okay, it contains a thiol group. S group is sulfur is there, nitrogen is there, that's why thiazolidine ring. This thiazolidine ring is condensed with the beta-lactam ring, and to the amino group variable R chain is present. Okay, there is R reflect variable chain, so variable groups can be present at this position. Okay, so different penicillin molecules differ in this region, different penicillin molecules contain different uh, groups here. Okay, so now what happens when the normal penicillin fermentation, when there is only bacteria and a culture broth and nothing is added to the fermentation mixture into the vessel, into the fermenter. Okay, there are only cells and there is only a culture broth. Okay, then natural penicillins are produced. Okay, so see, Penicillium chrysogenum naturally produced penicillin G. Okay, this is a natural penicillin. But when during the fermentation process different precursors are added, then different biosynthetic penicillins are produced. Okay, during fermentation process, if different precursors are added, different biosynthetic penicillins are produced. Okay, and when once the natural penicillin is produced, then if the natural penicillin is treated, chemically or enzymatically treated, then semi-synthetic penicillins are produced. Okay, what happens for the production of semi-synthetic penicillins? The natural penicillin G is treated to form, to give this 6-amino penicillinic acid, which is then added with different kinds of side chains chemically. Okay, there is chemical addition of different side chains, so different semi-synthetic penicillins are produced here. So ampicillin is produced, amoxicillin, methicillin. These are the different kinds of semi-synthetic penicillins which are very commonly used. So why semi-synthetic penicillins are produced? There's a difference between natural penicillins and semi-synthetic penicillins. Natural penicillins are narrow spectrum antibiotics and they can have more toxicity, whereas semi-synthetic penicillins, they are broad spectrum antibiotics and they have less toxicity. Apart from that, natural penicillins, they cannot be absorbed orally. They're not good when taken orally, because these are acid sensitive. Again, where we have a acid in the stomach, and natural penicillins, they get degraded with the acid. But semi-synthetic penicillins, they can be taken orally. There is no problem with their absorption. So these are some positive things. That's why semi-synthetic penicillins are produced. Coming over to production of penicillin. So penicillin G is produced in fermenters of 40,000 to 2 lakh liters capacity. So large fermenters are used because penicillin is used, very widely used. So it is like produced in large quantities. So large fermenters are used. So penicillin production is highly aerobic process and efficient aeration is critical. Now for the production of penicillin, the fungus which is used is of course that is aerobic. So the process is aerobic and efficient aeration is critical, because aeration is a problem for aerobic fermentation, because large fermenters are there, and in large fermentation, mixing of the air, because sterile air is passed through the fermenters, so mixing of the air in the huge fermenter is very critical, because all the cells in the fermenter, they need air, they need oxygen, because aerobic process hai. So aeration jo hai, that is always critical. Then penicillin is a typical secondary metabolite. As I told that antibiotics are the typical example of secondary metabolites. So penicillin is a typical secondary metabolite. That means it is produced in the end of the log phase, that is exponential phase, and mainly in the stationary phase. Okay, then the major ingredient of penicillin production media is a corn steep liquor, which is the source of nitrogen and growth factors. Okay, so for nitrogen and growth factor, corn steep liquor is used. Then importantly, high levels of glucose repress penicillin production, but high levels of lactose do not. So lactose is added to the corn steep liquor in large amounts as a carbon source. Okay, so this is clear. High levels of glucose repress penicillin production, so glucose is avoided to be used as a carbon source. Instead of glucose, lactose is used. Okay, so high levels of glucose repress penicillin production, but high levels of lactose do not. So lactose is used as a carbon source. Okay, then as the lactose becomes limiting and cell densities in the fermenter becomes very high, feedings with low levels of glucose maximize penicillin yield. Okay, so when the amount of lactose becomes less during the cell growth, during fermentation process, then during that time feedings with low levels of glucose are added, and this is added because this maximizes the penicillin production. So this come as a part of bioprocess. Okay, so this is something that scientists have standardized, that okay, when the lactose becomes limiting, then if they give small feedings of glucose, that maximizes penicillin yield. This we are going to understand with the help of a figure. So at the end of the production phase, cells are removed by filtration and the pH is made acidic so that no more penicillin is produced. Then penicillin can be extracted and concentrated into an organic solvent and finally crystallized. Now let us have a look on the production process of the penicillin. एक बार figure देख लेते हैं. Now let us have a look on the kinetics of the penicillin fermentation with Penicillium Chrysogenum. This figure shows the kinetics. So in the y-axis there is biomass, concentration of carbohydrates, ammonia and penicillin. On the x-axis there is a fermentation time. So in total the fermentation process takes around like more than 5 days. It is 140 hours, that is more than 5 days. Now the red line in the graph shows the cells, growth of the cells. The blue line shows lactose, black line shows ammonia, and the green line shows penicillin. Now as you can see, the blue line, as the lactose concentration goes down during the fermentation process, you can see here the small feedings of glucose. Small glucose feedings are given at different time intervals during the fermentation process, as well as nitrogen feedings are also given at different time intervals. If you plot this bar down to the time, you will see it around 50 hours, at around 50 or 45 or 50 hours or 45 hours, first glucose feeding was given. And as the feeding was given, you can see sharp increase in the concentration of penicillin. You can see sharp increase in the penicillin production. Then again at different times, the feedings are given for higher yield of penicillin. Okay, you can see from this graph. So this happens when lactose concentration goes down, the penicillin production goes up at this point. Okay, so here the penicillin is produced as cells are entering into the stationary phase. You can see if this is a growth curve, then this is a log phase, and then from here stationary phase starts. There's a bend in the curve. So penicillin production starts taking place when the cells enter the stationary phase. So from this point, from here, penicillin production starts taking place. Okay, when the cells are entering the stationary phase, at this where you can see the arrow. So this was about the kinetics of the penicillin fermentation. Let's move further. Now coming over to the production of tetracyclines. So tetracyclines are antibiotics having the four-membered naphthacene ring. These four rings together, these are called naphthacene ring. So tetracyclines are four-membered naphthacene ring. In chlor tetracycline, there is a chlorine atom at this position, and for doxycycline, there is a hydroxyl group at this position. Now, for the commercial production of tetracycline, chlor tetracycline, Streptomyces aureofaciens is used. Okay, Streptomyces aureofaciens is a strain which is used for commercial production of tetracyclines. For tetracycline synthesis, large number of enzymatic steps are required. It is a very tetracycline production is a complex metabolic process. So large number of enzymatic steps are required. For synthesis of chlor tetracycline, 72 intermediates are there in the metabolic process. So there are 72 intermediates and around 300 genes are involved in the production. So it is a very cumbersome process, complex metabolic process. Now, what are the key regulatory steps in the production scheme? For chlor tetracycline synthesis, it is repressed by both glucose and phosphate. Okay, both glucose and phosphate repress chlor tetracycline synthesis. So the medium used in commercial production contains very low phosphate concentration. Okay, so for fermentation process, low phosphate concentration is used. Then instead of glucose, sucrose is used as a carbon source, and corn steep liquor is used for nitrogen and growth factors. Now let us have a look on the production scheme of chlor tetracycline. Now this is the production scheme for chlor tetracycline using Streptomyces aureofaciens. So first of all, what happens in the scheme? First of all, the spores of Streptomyces aureofaciens are inoculated on the agar plate using the optimum medium, and this is the medium which they use. This is 2% meat extract, 0.05% asparagine, 1% glucose, 0.5% K2HPO4, and 1.3% agar. So by using this medium, spores are first inoculated on agar plates. Then the spores are then inoculated on in the shake flask. Shake flask having this medium. Okay, this medium, which is 2% corn steep liquor, 3% sucrose, and 0.5% calcium carbonate. Okay, so this is a medium which mimics the production medium. Okay, this medium mimics the production medium. That means this medium is similar to the medium which is used for final commercial production. Now in the culture flask, the spores are inoculated for 24 hours. Okay, then in the pre-fermenter, again the cells are then inoculated, and with this same medium, 2% corn steep liquor, 3% sucrose, and 0.5% calcium carbonate. So the same medium which mimics the production medium, in this medium cells are inoculated in a pre-fermenter. Okay, the pre-fermenter is a small fermenter. It is a lab fermenter. It has a small capacity like 5 liters or 10 liters capacity. Okay, so cells are grown in this medium for 19 hours to 24 hours, and pH is maintained at 5.2 to 6.2. Okay, this pH is maintained. Then the same process. No, this is a process. Okay, like medium 2% corn steep liquor, 3% sucrose, 0.5% calcium carbonate, incubated for 19 to 24 hours, pH maintained at 6.2 to 5.2 to 6.2. This is a process. Okay, now this process is scaled up to the large scale fermenter. Okay, after pre-fermenter, now the cells are inoculated in this medium. Okay, this medium in large fermenters from 60 to 65 hours. Okay, the fermentation process here takes place from 60 to 65 hours and pH is maintained from 5.8 to 6.0. Okay, so this medium is used. 1% sucrose, 1% corn steep liquor, 0.2% ammonium hydrogen phosphate, and 0.1% calcium carbonate, 0.025% magnesium sulfate, 0.005% zinc sulfate, 0.00033%, and each of copper sulfate and magnesium chloride. So this is the production medium, and this medium doesn't have glucose. Okay, this medium lack glucose, and also the phosphate concentration is also kept at very low, because glucose and phosphate represses the synthesis of chlortetracycline. Okay, the presence of glucose in the medium and high phosphate concentrations would repress the synthesis of chlortetracycline. So the production medium, even the small scale medium, here also sucrose is taken, not the glucose. Okay, and phosphate concentrations are also kept low. Okay, so now after 60 to 65 hours, antibiotic is purified from the broth and cells are removed, and you get your chlortetracycline here. Okay, that is purified. So this was about the production scheme. Now if you look at the medium which is used for growth of the spores on the agar plate initially, this medium, which is the optimum medium for the growth of the streptomyces or aureofaciens spores, this contains the glucose. This contains 1% glucose, but then in later steps glucose is not used. Glucose is replaced with sucrose. Okay, it is important for production of chlortetracycline. Okay, so I hope this is clear. That's all for today. If you have any doubt, if anything is not clear to you, or if you have any other question, you can write on the comment box, and see you in the next lecture.